under animal diversity 2, we shall talk about the general characters of chordates now. The chordates evolved during Cambrian period. They evolved during Cambrian period of Paleozoic era. The term chordata was coined by Balfour. The term chordata was coined by Balfour. Now the chordates exhibit highest rate of diversity, with the smallest animal to the largest animal like Balanopterus musculus. The fundamental characteristics of chordates are: number 1, notochord; number 2, dorsal tubular nerve cord; number 3, pharyngeal gills; number 4, post-anal tail. They are the 4 fundamental characters of chordates now. Among these, the notochord. The notochord lies on the mid-dorsal surface of the chordate embryo. It is unsegmented, solid structure. It arises from chorda mesodermal tissue. Chorda mesodermal tissue that originates from the mesoderm gives rise to notochord. Notochord is also called chorda dorsalis. Notochord acts as embryonic axial skeleton. It gives support to the embryo. At the same time, it provides surface for the muscles of the embryo. Now, it remains only in the embryonic stage, and in adults, it transforms into vertebral column. The remnants of notochord in mammals are known as nuclei pulposi. The notochord has transformed into vertebral column, but in mammals now, the leftover cells of notochord are seen on the intervertebral discs now. So those leftover cells are the residues of notochord present on the intervertebral discs are called nuclei pulposi. The same way, the proatlas is also supposed to be the remnant of notochord. Proatlas is present in sphenodon and crocodiles. Proatlas is present in a reptile called sphenodon and another group of reptilia called crocodilia. The notochord lies above elementary canal and below nerve cord. It is unsegmented, solid structure, extends throughout the length of the body now. Then it transforms into vertebral column. The chordates with the vertebral column, that is in which the notochord has transformed into vertebral column, are called vertebrates. The chordates in which the notochord is still persistent even in the adult stage are called protochordates. The second fundamental character of chordates is the presence of dorsal tubular nerve cord. This nerve cord is hollow, tubular, lies above the notochord, extends along the midventral surface of the body. In chordates, the nerve cord arises from neurectoderm. In chordates, nerve cord is single, it is hollow and it is non-ganglionated, whereas in non-chordates, it is double and solid and ganglionated. In all non-chordates, it is midventrally placed, whereas in chordates, it is mid-dorsally placed. In chordates, nerve cord arises from the neurectoderm by invagination, whereas in non-chordates, it arises from the ectodermal region by delamination. The nerve cord is hollow with a ventricle called neurocele, which is filled with cerebrospinal fluid, while in non-chordates, it is completely solid. The third fundamental character of chordates is post-anal tail. The posterior extension of the body that lies beyond the anus or cloaca is called post-anal tail. In chordates, it is solid due to extension of notochord in the embryonic stage and vertebral column in adults. and it acts as a balancing organ in some chordates, acts as organ of offense and defense in certain other chordates. The other characteristics, another characteristic of chordate is the pharyngeal gills. Pharyngeal gills. The ventrolateral perforations present in the pharyngeal region of chordates are called pharyngeal gills. The pharyngeal gills are formed by invagination of outer ectoderm and evagination of inner endoderm. That is, they are formed due to involvement of both ectoderm and endoderm of the pharyngeal region. The pharyngeal gills remain persistent only in certain chordates like fish and certain amphibians. In all other chordates, they are present only in the embryonic stage and disappear in adults. The pharyngeal gills help in respiration in certain amphibians and in all fish, whereas in urochordates, instead of respiration, they help in food capture. That's why the urochordates and cephalochordates and hemichordates, they are also called filter feeders, wherein the gills, instead of respiration, they help in food capture. Then the other fundamental characters of chordates apart from this are, number one, cephalization. Most of the chordates are cephalized animals. Most of the chordates are cephalized animals, so cephalization. Then all of them are bilaterally symmetrical animals. Bilaterally symmetrical animals. Then the segmentation, if you take segmentation, they exhibit only internal segmentation, external segmentation is absent. Vertebral column, that is vertebrae, and the spinal nerves, the muscles of the back, and the blood vessels in the embryonic stage, nephrons in the embryonic stage exhibit internal segmentation, and external segmentation is completely absent. Then if you look into the blood vascular system of chordates, in all chordates, heart is ventrally placed, and heart is myogenic. It may be 2 to 4 chambered. In all chordates, heart is myogenic, where the heartbeat is initiated by a muscular constriction called sinoatrial node. And in chordates, the blood vascular system is characterized by the presence of portal system besides the normal blood vessels. The portal system is again divisible into hepatic portal system and renal portal system. Presence of portal system is one of the unique characteristics of chordates. And in non-chordata, heart is dorsally placed, it is neurogenic, and the invertebrates do not have the portal system. In all chordates, hemoglobin is accommodated within the RBCs, whereas in non-chordates, hemoglobin is dissolved in the plasma. Then all chordates, the other characters of the chordates, I'll tell you. The other chordates, they exhibit radial and indeterminate cleavage. They exhibit radial and indeterminate cleavage. The eggs are equipotential. The eggs are equipotential. And all chordates are deuterostomians. All chordates are deuterostomians. And chordates, the phosphogen present in chordates is phosphocreatine. The phosphogen present in chordates is phosphocreatine. So these four are the fundamental characters, that is notochord, nerve cord, postantral tail and pharyngeal gills are the fundamental characters of chordates. All the other characteristics are the normal characters which are shared even by the invertebrates like cephalization, bilateral symmetry, segmentation, radial indeterminate cleavage. Then eggs are equipotential, deuterostomian nature, the phosphogen being phosphocreatine. Like in invertebrates only the annelida, arthropods are cephalized In rest of the invertebrates, like for instance you take Porifera, there is no cephalization. Cnidaria, there is no cephalization. Platyhelminthes cephalization is noticed for the first time. Okay, Nemathelminthes cephalization is indistinct here now. Annelida and Arthropoda is distinct. Once again, in mollusca also distinct cephalization is noticed in most of them. Echinodermata, there is no cephalization. Segmentation, invertebrata, they exhibit both external segmentation and internal segmentation, whereas chordates only internal segmentation is noticed. Then in chordates, cleavage is radial and indeterminate, whereas in non-chordates it is spiral and determinate. In chordata, eggs are equipotential, whereas in invertebrata they are mosaic. And invertebrata, most of them are protostomians. The only deuterostomian invertebrate phylum is Echinodermata. The phosphogen present in invertebrates is phosphoarginine, whereas in vertebrates it is phosphocreatine.